Good evening, everyone. Welcome to Botany Insiders new class. Let's wait for others to join in and then we'll start off with our class. We'll be discussing about the basics of Drosophila, which is one of the most important concept when you look into unit number 5, that is developmental biology. Okay. Let everyone join in and then we'll start off with our class. Good evening, Shiva. Good evening, Komal. Let others also join, then we'll start. Good evening, Lena. Okay, so let's begin with our class now. We'll be discussing about the developmental biology's Drosophila topic. Okay, before starting the video, before starting our class, make sure that if you are preparing for the December examination and you wish to have a structured preparation, you can enroll in our crash course, which is ongoing right now on our application. You can download our application from the iOS as well as from the Play Store. Okay. So you can download our application and then you can get yourself enrolled in this particular course. And we're about to start with the PYQ course for the December 2023. We'll be discussing all the previous year questions that are important from your exam point of view for the upcoming examination in the most detailed manner possible. Okay. So let's begin with our class now. Good evening to all the students. So we'll be discussing about Drosophila. It's one of the most important topic from unit number 5 point of view, that is your Def Bio. Okay. Or we can also call it as developmental biology. First thing first, we'll be discussing about what are the most important segments from this particular topic, and then we'll be continuing ahead. See, if you look into or if you analyze the previous year questions properly, you will see that almost every year, every single year, you can find a question from here. Okay. And what type of questions are asked? First, the type of questions are from the gene level. Like there are various type of genes that are there that are involved in the formation of the various segments that are involved in the number of the appendages and the flies, halters, all of these things. So how do these things get formed? You get a question from the gene level. You'll be getting a complete list. Like when you'll prepare, you'll see that there's a complete list of the genes that are involved. Then you get questions from this particular unit in this particular topic, specifically from the experimental point of view, that is in relation or with unit number 8 and unit number 5 combined, that is your genetics and your Drosophila combined, you get the questions from this unit. Then in addition to that, you also get questions like sometimes directly statement wise questions can also be asked. In addition to that, at gene level, you might get questions in the match the following format. And then you also get questions from mutations. Okay. If a specific gene get mutated, how will the final body segmentation get impacted, or what are the various backfalls or drawbacks that are there if by chance any mutation occurs. So all in all, this entire topic is very important. Is this particular point clear to all? Like if you all have observed like the previous year questions properly, you also have understand that how important this particular topic is, right? Just understand this and then we'll proceed ahead with understanding what is Drosophila's basic mechanism, or we can say the basic characteristic features, then what or what is the life cycle of Drosophila all about, etc., etc. Just go through this, everyone, then we'll proceed ahead. Understood, everyone, till here? Okay, let's proceed. Hello, Vagisha. Hello, Kamyani. Okay, let's proceed. Right. Very first thing, write down in the chat section what is the common name for Drosophila. Everyone, write down the common name for Drosophila. What is it? Drosophila's common name. Fruit fly, right? Or we also call it as a common fly, right? It is the most common name is fruit fly. Okay. Now understand this over here. So these are like every single point that you'll see in the slides is important. Okay. So what is Drosophila all about? So the common name is it's a fruit fly. Then why do we have to study Drosophila? Reason being, it is like the model organism in the entire developmental biology. Okay, what is it? It's the model organism in the entire developmental biology. Reason being, studying this particular fly, you get to learn a lot of factors, a lot of important mechanisms which helps in making or getting the deductions done for various other organisms as well. Okay, so it's used in the research in the genetics as well. In addition, it's a model organism basically, or the like majorly in the developmental biology. Then over here, if you look into the body type, so the males have the Drosophila males have the longest sperm of any organism. Like on the earth, the longest sperm are found for Drosophila. Then the what is the lifespan? Like see, because it's a model organism. So that's the reason why, or the reason for it to be considered as a model organism is because it have a very short lifespan, and that is maximum 21 days. Reason being, from the egg stage till the death of this particular Drosophila fruit fly, the complete time or the complete lifespan is 21 days only. So it becomes easier for the researchers to cultivate them, to grow them, and then to study and then get a new cycle or a complete new fresh set of Drosophilas grown in culture. Okay, so that's the reason why it is known as, or it is the main developmental organism in your main model organism in the developmental biology. Then in addition to that, see, 21 days is the maximum time period. Okay, now this time period varies. This can vary depending upon what temperature is there. Okay, what temperature, or what is the temperature in which the Drosophilas are getting grown. So with the help of the same, you can get to, or there's a difference in the lifespan. So the shortest lifespan or the shortest development time is 7 days, which is at 28 degrees Celsius. They'll be having the shortest time or the shortest lifespan of the entire, like their complete lifespan it gets completed, or their complete uh, life cycle gets done within 7 days. Okay, so the shortest developmental time is 7 days. Every single point written down is important. First, the common name is fruit fly. Second, it's a development model organism in deaf bio. Then it's used in the research. Males of the male Drosophila have the uh, highest or the most longest sperm. Then the lifespan is 21 days. The lifespan varies with the temperature. This is a very, very important statement. Okay, remember about this particular statement. You can get this thing in the in your questions. You can easily get this particular thing. Then shortest developmental time is 7 days. Is everything clear to everyone till this particular point? Yes or no? Then we'll proceed further. Yes, clear everyone till here? Any doubt? Any problem? Yes, it is considered as the Cinderella of genetics, right? Let's move forward. Now you have that why is it considered as a model organism? Very important. Okay. In examination, you can expect a question from here in statement wise. Okay. In the statement wise thing, you can easily get a question. Very first and the most important pointer is that the genome of the Drosophila can easily be manipulated, and you have the manipulation and the investigation becomes very easy. Like the genome can be easily manipulated and the investigation of specific genes can be made in the Drosophila. That's why it is considered model organism. Then the genome size of the Drosophila is very small. Okay, it have a very small genome size, so it becomes easier for the researchers to read it, to understand it, and to investigate the differences. Then they have a shortest lifespan. So we have just understood new flies can be easily quick and they're they can easily be made and they are very, very easy to form as well. Then new lives take only 6 weeks to form. So it requires very less cost. In the experiments, the cost that is incurred is also taken into consideration to a great level. That's why the cost of uh, developing the organism is very less. That's why uh, these are considered model organism. And then last point, very important, that we have discussed initially as well. They have very common genome. So in case of your like 75% of the known diseases, they have a gene which matches with the fruit fly genome. So we can easily study a lot of diseases. We can easily learn about a lot of diseases, and then the treatment identification, or we can say learning the behavior of that particular disease becomes very, very easy for the investigators. Remember, it's a very important concept. Okay, so understand that the model organism, we are calling it, that's for sure. But what is the reason for that? So these all are the major reasons why we are calling it a model organism. First one, the genome can easily be manipulated. See, what is a model organism all about? A model organism specifically means an organism which can easily be handled, which can easily be available, and then which is very the it should be cost effective as well. That's the reason why we are considering or we are taking Drosophila. Reason being, it's a very common uh, organism first. Then you can easily cultivate them, you can easily study them, you can manipulate their genome easily, You can learn about a lot of diseases, learning about the, like when you learn about the Drosophila's entire genome, you get to learn about a lot of diseases because they share a number of, like a very good similarities there between the two. Is it clear to all? Yes or no? This is a very, very potential statement. Okay, it's a very potential statement. So remember about this. If this is clear, then we'll proceed further. Okay. Understood, everyone? Right, and if you want to learn in depth, in detail about Drosophila and all the other topics as well, we have a full course and we have a crash course going on right now. You can enroll in them. We have a very, very minimal pricing kept for the students. So you can enroll in that and get yourself that structured preparation. Okay, let's proceed further. Now look into the next part. Okay, let's understand about the next part, that is the complete developmental stage, or we can say what is the entire life cycle of your Drosophila. Very important. This holometabolous method of development has been asked in the examination previously as well. So learn about this. Keep a note of this particular thing. It's a very important concept. First thing first, what do you mean by holometabolous? Holometabolous also means complete metamorphogenesis. What do you mean by metamorphogenesis? So, morphogenesis means, morpho means morphology, genesis means synthesis. Metamorphogenesis or complete metamorphogenesis means that from first stage of your initial body type, or we can say the initial structure of the body, it completely reverts or it gets completely changed into an entire different type or an entire different complete uh, form is there in the final or the more adult form of the uh, Drosophila. Okay, so holometabolous means there's a complete metamorphogenesis occurring in the body. Okay, so what do you mean by holometabolous? So it means complete metamorphogenesis, and there are four stages in total. Okay, how many stages are there? So in the entire metamorphogenesis, you find four stages. What are the four stages? First, you have the egg, then you have the larva, then you have the pupa, then you have the imago or adult. This imago word have also been asked in the examination. Remember, adult or imago means the same thing. There are four stages. First, you have the fertilized egg. After that, you have larva, then you have pupa, then you have the adult or you have the imago, IMAGO. Okay, remember about this. This has been asked in the examination. So, holometabolous method simply means that there are four different stages of complete life cycle. What are the four different stages? Egg, larva, pupa, and imago. Okay. Remember about this. Two questions have been asked previously, specifically from this slide, and you can have a number of different other questions also framed. So keep a note of it. What is the type of the life cycle? What is the type of the developmental type are they showing? And what are the various four stages? Go through this, everyone, and then we'll be proceeding further. Very, very important. Okay. If you want to learn about Drosophila and if you want me to continue this particular topic on the YouTube channel, do let me know in the comment sections below. I'll prepare or we'll arrange the classes accordingly. If you feel like that you want to learn about this entire thing in detail, do let us know about it. Okay. And just go through this, and if you find any difficulty, you can ask. Understood, everyone? Yes, correct, Anjali. Butterflies and moths are also holometabolists because they show complete metamorphogenesis. Very good. Okay, sure. Others also do let me know about it because we have to arrange the classes accordingly. So, whatever is most suitable for you people, you can let us know. We'll try to provide you, like, we'll try to arrange the classes in similar manner. Okay, understood, everyone. Let's proceed further. Right, so this is about the four stages which you have to keep in mind. In addition to that, the next thing is related to the various genes. So till now we have understood about that what is Drosophila all about. Then you have understood about the life cycle. See, you'll have to learn in depth also because I'm trying to give you the best, like the most detailed manner over here. But obviously, because of time constraint, we cannot go in the most detailed manner. Reason being, uh, we have various other sessions also on our YouTube. So if you wish to learn it in the complete depth, you can come on the YouTube, come in the application. And if you want to just get through the baselines and understand the scenario, you can come in this particular session as well. Okay. Now the next segment, most important segment, is related to the genes. That is, what are the various genes that are involved in. So there are there are three main group of genes that we have that play the most important process, or that play a developmental role in the Drosophila. So first you have the first group of genes are the maternal genes. Then the second group of genes are the zygotic genes, and the third group of genes are your homeotic selector genes. Okay, so which are the genes? First one is the maternal genes, then you have zygotic genes, then you have the homeotic selector genes. In case of your zygotic genes, you have internally three segments of the genes. What are these? You have gap genes, pair rule genes, and the segment polarity genes. Okay, so maternal genes, zygotic genes, and homeotic selector genes. In case of your zygotic genes, first one are gap genes, pair rule genes, and segment polarity genes. Okay, every single gene will work in this sequence itself. First, the maternal genes will get activated. You have a number of genes in each one of the group, each of the group, and you'll have to keep a note of all of them. For example, in case of your maternal genes, there are genes, the name of the genes are, you have bicoid, you have hunchback, you have nanos, you have chordal, etc., etc. Like there are a number of other genes as well, but majorly there are four genes that play the role: bicoid, hunchback, nanos, and chordal. Then you have zygotic genes. Now, first, your maternal genes will get activated. All these things works on the basis of your morphogenic gradient. What are morphogenic gradient? So morphogenic gradient specifically means these are the chemical compounds that are there, which based upon their concentration gradient they are working in. If the concentration gradient is higher, they'll be performing more role. If the concentration gradient is lower, they'll be performing lesser role. So based upon the concentration gradient of the various genes that we have, now all of these are your like these genes will be activating a number of transcription factors. So depending upon the level of these genes, depending upon the level of the expression these particular genes will be showcasing, in the similar manner you will be looking into the functionality, you will be looking into the characteristic features that we have, right? So remember about this particular thing. Then you have zygotic genes. In the zygotic genes also, first the gap genes will be getting activated, then pair rule, and then the segment polarity genes. And then segment polarity genes will be activating your homeotic selector genes. In the homeotic selector genes also, you have two main groups of the genes that we have. So ultrabithorax is there, then you have antennaepida, then you have ultrabithorax 1, ultrabithorax 2, etc., etc. So there's a complete list of 22 to 25 genes, and every single question, like from every year, you can find a question in the from the gene level as well. Okay. Yes, but sure. So what are morphogenic gradients? So these are the chemicals that work based upon the concentration gradient. Okay, so what do you mean by morphogenic gradient? So morphogenic gradient or morphogens are those chemicals that work based upon the concentration gradient. So if there's higher concentration of bicoid, let's say over here, and there's lesser concentration of bicoid on the other side, so bicoid will be working more, or it will be showcasing more expression in the area where there is higher concentration of the gene present. Okay, so this is what morphogenic gradient are. Okay, clear everyone now? Is everything clear till this particular pointer? Go through this particular point, everyone. 'Yes'. Everything clear to everyone till this point? 20 of you are there. Make sure that you are getting your doubts cleared, because this is the point till, like, till here we'll be doing it today, and tomorrow we'll be doing from this particular segment, that is from the genes point of view. Okay, I hope till here things are clear, and if you want us to continue this topic, and if you want us to take up any other topic also, you can let us know in the chat sections. We'll work accordingly for you all, so that you can, like, prepare well and perform well in the examination. In addition, make sure to download our application and also hit the subscribe button to Botany Insider. I'm trying to provide you the best content possible, so share it with your friends as much as possible, and do let them know about our channel, okay, and our application as well. I hope it's clear to all. Let's meet in the next class now. All the best, good luck, take care everyone. Okay, bye everyone, take care.